Good morning, dear students. So this is a continuation of the last class, and we are going to perform the lead molecule search. For that, the step one is selection of biological target, right? So anyway, we are going to select a protein which should has the UniProt ID. If you have UniProt ID, means that's okay, unless you can model the protein. Otherwise, if you have only the sequence, okay, nice. As per your suggestion, you have given me SAC2 as a target protein. Instead of searching for human protein, I can take coronavirus target itself, right? I'm going to search what can we do in the coronavirus targets. Yes, because we already have the drug called remdesivir against SARS-CoV-19. So I'm going to search that target of that remdesivir so that we can get the standard target and the molecule both. Okay, so I will show you how to do it by using therapeutic drug target. And this is what is this? This is therapeutic drug target. Mm, what's happening? Maybe it's slow. Okay, and here I will give actually in this place we can give the target name, and here I am giving the drug name, right? So I'm going to search the target. If you have the target, you can use. Otherwise, if you have the drug, you can use. For your case, you already have a protein of interest for the throughout practical, so you can use that protein also. Otherwise, you can choose if you are not want to work on that protein means you can choose any pathogenic protein to target that. Okay, I will search here and see that if you want more information about this target means you can click here and you can see that in this practical purpose I need only the name of the target, right? So I can copy, see that of them. I just copy, otherwise I can copy full, no props. I can go for Uniprot. You can just paste here and search. See that there are so many proteins are available. You have to check with the organism, is it correct? Whether our results are we going to take the correct protein from the particular organism or not? Because I'm searching for COVID, so SARS-CoV-2 is here, right? Okay, I will choose that. I need the protein UniProt ID only, right? So I can just copy what I have to do. What is the second step? So now we have the target protein and we have the UniProt ID also. So next thing is you should go to pubchem and chembl, right? So I will show you how to do in pubchem first. I have copy here and this is pubchem database. You can get chemicals here, all the structure. See that remedies are yesterday I have searched, I think. So I will go to home now. Oh my God, what is the pubchem? See that this is the web page of pubchem. You can just paste here the protein ID, UniProt ID, and go to search. Don't use protein name as common one because there are so many sequences will be available. So it's better you can use the UniProt ID, right? If you don't get UniProt ID means you can use the sequence or otherwise you can use the name. If you have UniProt ID means it is better to use this. I will search it and see the next one. There are bioassays, 680 bioassays on genome protein, taxonomy, 31 pathways, and one literature is available. So this is the protein, severe acute respiratory syndrome, coronavirus 2, ORF1A. So I will go here, just click, and I can search bioassays. See that you want to go, you can go that, search bioassay. And in that bioassay, we need, you can see so many researchers, so many bioassays compounds with 7174 total substances are said, and here 924 active substances are found, and here active substance 581. These are the bioassays they have already submitted by the research labs, right, or companies. So you can just uh, select anyone if you want. Is there, right? You can go, that is biochemical assay from US patent SARS-CoV. If you want, you can select here. Otherwise, I'm going to select any one of these two, right? And here, just I will select here. What is the condition? And see that we need IC50 and IC50 values to select the best compounds, better compounds, right? So, what is it? In here, in this, I need that data table, right? The right hand side, you can just click on, otherwise you scroll down and see that. And here there are substances and IDs, activities are unspecified. There is no score given. Standard type is inhibition, standard relation equal to standard value. These are the values, drug values of in the um, micrometer, I think, sorry, nanomole, right? Converted into nanomole. And it's already in millimolar concentrations converted into nanomolar concentrations. Actually, here there is no detailed value, so I will skip this. I will check with another one. So I will go here, open the explore the potential non-covalent SARS-CoV-2 main protease inhibitors. Okay, so I just click here and I will get the data tables. So I need bioassay, right? Data table is here. So you can find here the activity is active and score, and here channel type is IC50. So all of them are inhibiting, right? The pubchem standard value, this is the converted value from the IC50. See that 0.065 and this is 0.00065 is converted. So we can download this, right? So we can go here and activity. I need the ascending, right? Because whatever you want, you can download. Don't worry. Anyway, we are going to process the Excel file. So download a CSV file. Here you can select, if suppose if you want to filter here itself means you can use activity less than or equal to 1 nanomolar or less than or equal to 1 micromolar or inactive substances. And here you can download all the data tables, means both the active inactive compounds, right? So I need active compounds, right? Data table, active compounds. So CSV, I will. If you want, you can go for activity uh, less than or equal to 1 micromolar. I already told you IC50 value should be less than or equal to 10 micromolar, right? That will be good. And it is better to go for nanomolar also, right? So here for the practical session, I will choose this active compounds. So I will download here. See that downloaded. You can open with the Microsoft Excel, so you can get that, right? So pubchem is ID here also smile structure. This is a smile structure of the compound active. All are active, right? We have downloaded all the active compounds. Okay, so we just we can delete all the things. Okay, this is standard type IC50. This is standard value. This is IC50 value, right? So we need this thing, and this is what the protein ID target we have given. So I don't want this thing. Okay, just delete. I can shift the cells. Okay, this thing also I don't want. Result active concentration and true. Okay, we can delete this also. See, vocabulary. This is smile structure and this is standard type. And outcome standard value, drug activity value, IC50 value, and here is the real IC50 value in micromolar, right? And this is in nanomolar. Is it okay? Right. So I will select this. You can go freeze it. Where is the freeze time? Oh my God. Select selection of time is there. That. Okay, leave it. I want to freeze that. Is there any freeze the first column, first row? Anyway, we'll make it as an okay. And here you can. We are in the need of this chemical smell structures. But before that, we want to filter by using this IC50 value, and we can sort, expand the sort by using IC50. What is that IC50? Values hardest, smallest to largest. Okay, so this is the thing. See that this compound has point 0.65, right? And that is the micromolar. Micromolar and this is nanomolar. Okay. So I told you less than or equal to 10 micromolar will be better. Yes. So and this is 3 and this is 20. Actually, we want to choose that relation micromolar. We say 50 values are in micromolars. Okay. So up to 20 we can, up to 10 we can choose. Okay. So this is the another thing. Make this table. Okay, like that. We are going to. I will save the file. Where is bi? Okay, we'll save here itself. Pubchem layer. Okay, I will save as Excel. It's better. BI lab. I have saved that. The same thing you should uh, do in the chembl, right? You can just go to chembl and just put here the Uniprot ID, right? I can search it. We need targets, right? So you can just hit here the target. What happens? 19 targets are there. And just go for the view, different view. Okay, you can just click the table view, and in the left hand side panel you can choose anything if you want, right? Just go through that. You can use that. Okay. We are in need of activity. What is where is the bio say activities? Organism compound BODs or single protein. Okay. So sometimes that's why we should give the Uniprot ID. Otherwise, you may get a different proteins. So here you can select only assay format histogram. Sometimes for this protein and here Homo sapiens coronavirus syndrome. Sorry, no need. I need targets. It is taking time to load. Chemical ID name any product session type single protein. We need single protein. Okay. Protein complex again single protein complex. Okay. How many actually we should choose how many compounds and activities, right? There are this much activities how find replicase polyprotein lab, right? You can go for this compound. So just I will, if you don't find, if you cannot find this molecule means just you can give search and you can find that. Okay. organism, and also what is that? This is from minimal, right? We need a single protein with the highest activity. For this practical purpose, I will choose I will choose here and this one. Okay. So the structures are available for you with the IC50 values, estimated standard values, right? So you can download as a CSV file. Why it is happening? Dismiss. Okay. I think there are 20 pages. It will take time to download. So if you want, you can filter that. See that there are compounds, approved compounds, this many, and clinical trial phase 1, phase 2, phase 3 are available. In preclinical, there are 9,603 compounds are here. So this very big library. That's why it is taking time, I think. So it's better we will go for K value. So inhibition, inhibition compounds, and here target organism. Okay, molecular weight we will do in Swiss ADME, and okay, this is how many pages? 10, 20 pages, 9526 records. Okay, last. It's taking time to download. So by this way, you can download like a pubchem. You can download and process based on the standard value, right? I told you less than 10 micromolar will be better. And here this is 9.4. Likewise, you have to find which are all things with less than 10 micromolar, less than or equal to 10 micromolar. Okay, in the Excel file, right? CSV file.